Hello and good morning, I guess, to all our colleagues in the US, and good afternoon to everyone here in Europe. So I'm today's host of this seminar series, and it's a pleasure for me to introduce Claire Deo, who will be giving this joint optical interest group seminar. And so Claire is a chemist by training. She studied actually photoswitchable complexes during her PhD at the École Normale Supérieure in Paris. And I guess for many Janelians, they will still remember her time as a postdoc in the group of Luke Lewis, where she developed bright and redshifted calcium indicators based on the Janelia fluoro dyes. And so since last year, or 2019, we are very happy that she has joined EMBL Heidelberg, where she's now leading her own group with the aim to develop next generation fluorescent probes for biological imaging. And this is exactly the topic that I guess she will be speaking to you about today. And we are very much looking forward to your presentation, Claire. But just before we get started, I also remember I should encourage everyone to ask questions after the talk, but not by using the chat, but by rather using the raised hands. And then please unmute yourself and share the video. And hopefully by doing this, we can get nice interaction between the two institutes and all the people here. So with this, I hand over to Claire. Please, Claire, the screen is yours. We're looking forward to your presentation.
Thank you, Robert. I'll just share my screen now. How's it looking? Good? Cool.
Yeah.
So great. Thank you very much. So yeah, thank you very much for the introduction, and thank you very much for having me speak today at this joint seminar series. As currently at EMBL and formerly Janelia, I'm really excited to be speaking in front of both of the crowds today. And today I'm going to present some of my former work that I did actually at Janelia and talking about the development of hybrid molecular reporters for biological imaging. So fluorescence imaging has become the modality of choice to investigate biological systems. And as the biological questions become increasingly more complex, there is a critical need for developing new molecular tools that are really tailored for certain specific applications. So the fluorophores that are commonly used for biological imaging can be divided into two main categories. So on one hand, we have synthetic small molecules. Wait, I'll put my pointer maybe. Yes, can you see it? Yeah. So on one hand, we have the synthetic small molecule dyes that are made chemically. They have been engineered for 100 years. They have very high brightness, good photostability, and benefit from the structural diversity that is allowed by synthetic chemistry. And on the other hand of the spectrum, we have fluorescent proteins, which have the very interesting benefit of being genetically encoded and can therefore be expressed as fusions of protein of interest within cellular system, which has made their success in fluorescence microscopy in living cells and organisms. However, fluorescent proteins also have their own limitation. To illustrate this is just a graph representing monomeric fluorescent protein plotted by their brightness as a function of their emission wavelengths. And in the green region of the spectra, fluorescent protein perform really well and show really good brightness. However, this decreases really fast as we move towards more redshifted areas of the spectrum. So fluorescent proteins often show limited brightness at longer wavelengths. In addition, they also have other limitations, such as often reduced photostability in comparison, pH sensitivity, dependency on oxygen for maturation. And as a comparison with that, this is just a subset of commercially available synthetic fluorophores. And we can see that in terms of brightness, they clearly outperform available fluorescent protein, not only over the full visible range, but even in the far red regions, and can reach wavelengths that are still not accessible with the protein-based scaffold. So due to this limitation, there has been a growing interest in utilizing the properties of the synthetic small molecules for advanced microscopy applications. However, small molecules by themselves do not contain the necessary specificity to label features of interest. The answer to this problem started emerging about 15 years ago with the beginning of the hybrid fluorescent labels and the self-labeling tags, which are protein that can be used and expressed in a similar fashion as fluorescent protein, but are constitutively non-fluorescent. They do have the ability to recognize and bind specifically to a small synthetic ligand, which can then be attached to diverse fluorophores. And this allowed to benefit both from the genetic specificity of the protein scaffold and taking advantage of the brightness, photostability, and all the diversity of the developed synthetic dyes. So this was first developed with the SNAP tag, which is a 20 kilodalton protein that can recognize this SNAP tag ligand. And then a bit later on with the slightly bigger halo tag protein, which can bind to this chloroalkyne ligand. So these self-labeling tags are highly specific for their functional ligand. They're orthogonal, show fast binding kinetics, and additionally, their ligands are cell permeant, which allow them to be used in living cells. They can then be attached to a variety of diverse small molecules and functional handles, and in particular to fluorophores. And although in principle, this recognition is generic, they have been particularly well engineered for rhodamine type dyes. Rhodamines are among the most widely used fluorophore for lifestyle imaging. They have a very simple structure and have been enhanced and optimized to develop bright small molecules that are cell permeant and can also be very easily functionalized. And this effort has been made by, among other, the group of Luc Levis, and it has been shown that by simple structural modification, rhodamines could be made to maintain these interesting properties and cover the full spectral range to be used in imaging experiments. In addition, rhodamine have this very interesting properties of undergoing an environment-sensitive open-close equilibrium between a form that is completely closed, that shows no absorption and no fluorescence, and a form that strongly absorbs and fluoresces. And this equilibrium is strongly environment-sensitive. And this has been used for the development of so-called fluorogenic dyes that can turn on upon binding to their target. So this was first evidenced with the silicon rhodamine, the far-red silicon rhodamine halo tag ligand, which in solution is mostly shifted towards its closed form. However, upon binding to the halo tag protein, a fluorescence increase of about 7-fold is observed. And this is due to the shift of the open-close equilibrium upon binding towards the more open form. The exact basis of this increase is not exactly known, but what we can see from the crystal structure is that it evidently originates from the very tight interaction of the dye on the protein surface and the change of environment elicited by the protein surface residue. Following this observation, some structural modifications were there implemented into fluorophore to try to really increase this fluorescence increase and this property by introducing different functional groups on this diverse rhodamine. And this was extended to far-red silicon rhodamine such as Janelia fluor 635 that shows more than 100-fold fluorescence increase upon binding to halo tag, and then was also adapted through other synthetic methods to a diverse multicolor rhodamine, showing increase in fluorescence as high as 1,000-fold upon binding. So all this development of rhodamine and small molecule has made the use of rhodamine and their tags a very robust labeling platform with bright photostable dye that can be adapted to multicolor labels and that have been used for a diverse system, so they can be used in combination for multicolor imaging in cells. This high fluorogenicity property allows them to be used in no-wash imaging experiments, and they can also be used in awake animals. So during my postdoc at Janelia, what I worked on is to develop methods to use this rhodamine tag platform as a general methods to engineer functional reporters. And And the group of Luke Lavis, and then in collaboration with Eric Schreider, we worked on the development of sensors for calcium, because calcium is a very important second messenger involved in a variety of different pathways. And monitoring calcium can serve as an excellent proxy for neural activity. So it's particularly important in neuroscience and neurobiology. The main indicator for calcium that is used today is this family of sensors called GCAMP. GCAMP, which Jan talked about during her last joint seminar talk. So GCAMP is a genetically encoded calcium indicator, relying on the use of a GFP derivative attached to a calcium binding domain. And upon calcium binding, the change of environment around the GFP chromophore results in a large fluorescence increase. The latest versions of GCAMP, GCAMP 6S, and now even more with the new 8 improved variant, showed really good brightness and a very high fluorescence increase upon binding calcium, and are very robust sensors for monitoring calcium. However, the effort in engineering redshifted variant based on the same approach by exchanging GFP with more redshifted fluorescent protein resulted only so far in sensor with much reduced mass, but also much reduced sensitivity. And this is due, as I was first showing, first to the reduced brightness of the far-red fluorescent proteins themselves, but also illustrates the difficulty in engineering this protein that have more complex chromophores. So at Janelia, I worked on 2 different approaches to engineer a new type of redshifted fluorescent calcium indicator. In the first approach, we designed synthetic calcium indicator that we then used halo tag as a targeting motif. And then in the second approach, we used this time a genetic sensing domain to develop a hybrid system more analog to GCAMP that I worked on in collaboration with Eric Schreider. So for this first approach, we set out to develop redshifted synthetic calcium indicators. For this purpose, we based our design very similarly to what has been done in the past for a synthetic calcium indicator by attaching a small molecule fluorophore with the BAPTA calcium chelating motif that was developed by Roger Chen. And in the absence of calcium, the fluorescence of the fluorophore is quenched through a photoinduced electron transfer. However, upon binding calcium, this effect is much reduced, which recovers the fluorescence of the fluorophore. And this molecule is Oregon green BAPTA, which shows a high quantum yield and a fluorescence increase of about 14-fold upon binding calcium. So to develop similar platforms redshifted, we started our work with the bright Janelia fluor 549 rhodamine that was previously developed in the Lavis lab. We first set out to introduce the BAPTA chelating moiety on the bottom ring, similarly to what has been done for Oregon green BAPTA. And this approach gives a compound with a fairly high quantum yield, however, a limited dynamic range. We then set out to systematically evaluate how the relative position of the fluorophore and the BAPTA would affect the sensor property. So if we synthetically move the BAPTA and bring it one step closer to the fluorophore, the very high quantum yield is maintained, and we observe an increase in sensitivity. So we systematically explored this by introducing the BAPTA at the 7th position, and then further utilizing the azetidine ring as a new handle for functionalization. And what we have observed is that the closer the BAPTA gets to the fluorophore, the more sensitive the compound gets. However, if we get too close, such as in this compound or this one, it severely decreases the quantum yield of the fluorophore. So through this observation, we identified that positioning the BAPTA on this azetidine ring was what was giving us the compound with the best properties, which are properties that are slightly higher than Oregon green BAPTA and redshifted by about 50 nanometers. We then set out to evaluate the use of this compound in a cellular context for monitoring calcium concentration. So we designed a cell permeant version of it, and that we then delivered to culture neurons. And what we observe is that when this compound is by itself delivered into neurons, it tends to accumulate inside the nuclei of the cells. So to circumvent this natural and uncontrollable accumulation of the sensor, we use the halo tag as the targeting motif. And the benefit of using the azacitidine handle to introduce the BAPTA is that it also leaves the bottom ring of the rhodamine open for a functionalization with this halo tag ligand at the position which has been optimized for halo tag development. And we observed that this compound was still responsive to calcium in a similar fashion, and that binding to the halo tag did not disrupt the calcium sensitivity. When we use this compound in neurons that are expressing cytosolic halo tag, what we observe is that this compound does label especially the halo tag. And as we can see, there is no observable signal from the cell nuclei, therefore showing that this kind of intrinsic localization can be circumvented by using the halo tag targeting method. And this compound is sensitive to calcium also in cells with a delta F over F of about 10% for a single action potential. And in comparison in cells to the genetically encoded calcium indicators that are emitting at the same wavelengths, this compound is in cells substantially brighter. We then extended this approach to much rather fluorophores. So swapped in our design the rhodamine with the corresponding silicon rhodamine, Janelia fluorescent 646, that emits around 65 nanometers. And this compound shows very similar behavior to the JF549 derivative with similar localization and similar sensitivity, showing that shifting the wavelengths does not affect sensor properties in cells. To demonstrate the usability of this compound to intracellular localization, Hughes in David Clapham's lab evaluated its use to monitor calcium in the primary cilium, which is a very compartment. So as it has a very small volume, it requires the use of a quite bright indicator to visualize sufficient signal. And this compound targeted to the cilium does show a fluorescence increase homogeneously across the entire cilium upon calcium encaging. So in this first part, we've shown that we could use the halo tag as a targeting motif to localize specifically synthetically constructed calcium indicator. This approach works. There is still some limitations that come with it. First, as the calcium binding part is completely synthetic, the resulting molecule is fairly large. And secondly, as the entire calcium sensor is in one part, the sensor is still functional when not bound to the halo tag target. So if there is mislocalization in certain environments, this can lead to certain non-specific labeling and signal. In parallel, I also worked with Eric Schreider on engineering a second type of far-right calcium indicator based on a different approach in which this time, instead of having the calcium sensing part synthetic, has the calcium sensing domain genetically encoded and fused to the halo tag. The benefit of this approach is that it requires only the delivery of a much smaller molecules. And secondly, that as the sensor is separated into these two parts, the sensor becomes functional only where the ligand is tag bound, which provides a higher specificity as well. And this approach relies on the exploitation really of this open-close equilibrium of the rhodamine that I was presenting earlier, and that is highly environment sensitive. The design of this sensor paralleled the developments of the GCAM calcium sensor, which is this green fluorescent protein derivative attached with the calmodulin and calmodulin binding peptide. And as I mentioned, the way the sensor works is that upon binding calcium, the change around the environment sensitive chromophore of the GFP results in this fluorescent change. So the reasoning was that we could engineer a similar platform to GCAM by replacing the environment-sensitive fluorophore included in GFP by a rhodamine environment-sensitive fluorophore attached to the Halotag platform. And for this, we used the Janelia Fluor 635 as the far red chromophore, and engineered the Halotag by structure guided by the crystal structure by identifying positions in close proximity to the chromophore where we could engineer the Halotag to introduce sensing domain. And with this, we identified that position 143 in this loop facing the silicon rhodamine was amenable to engineering and functionalization without affecting the binding property of the Halotag platform. So we engineered a couple of different variants of this platform by using different camodeline binding peptide, and this resulted in 2 different variants with the dye Janelia Fluor 635. Both of these variants show high brightness with the delta F over F of about 5 for our first variant that shows a brightness of 75. So as a reference, this is about 1.7-fold brighter than GCamp 6S and redshifted by 140 nanometers. And this is also close to 20-fold brighter than the far-red fully genetically encoded sensor near Gecko 1. And we also engineered a second variant based on a different camodeline binding peptide that is a bit dimmer, but more sensitive. And that also shows a higher affinity for calcium. So we can measure the spectroscopic property in vitro. And what we can see for this first variant is that upon binding calcium, we see this large increase in both the fluorescence, but also the increase in the absorption. So seeing that it is really an absorption modulation driving process does demonstrate that the mechanism by which the sensor works is really a modulation of its open-close equilibrium that drive the sensor. And here we can see the titration curves for the 2 different variants. We solved the crystal structure of HaloCamp bound to Janelia Fluor 635 for one of these variants. And you can see here the insertion of the calcium binding domain on position 143 with the rhodamine dye sitting here on top of the cavity of the Halotag and facing outward. So this demonstrates that this approach works for the design of far-red calcium sensor. One benefit of this design is that different rhodamine dyes can be used with the same protein platform. So if we just swap and replace the far-red JF635 by other fluorogenic Janelia Fluor dyes, we can have a sensor that also performs with the emission around 600 or with emission around 540 nanometers. So the color of the sensor can be changed and adapted only by changing the small molecule fluorophores that is used. And here we can see that the sensor does not work with the tetramethyl rhodamine, which is a fully open rhodamine. And this also, again, shows that if the dye is fully open, there is no further modulation of this open-close equilibrium. In addition to the color variation and tunability of the sensor, we can also, through synthetic chemistry, very finely tune the properties of the sensor. So I synthesized a different series of the far-red silicon rhodamine dyes, so bearing different substitution groups on the rhodamine handles. And this does not change substantially the spectral properties, so all these dyes can be excited in the same region, but this does slightly change the open-close equilibrium of the dye, with a more electron withdrawing group shifting it towards the more closed form. This does affect sensor property and allows to fine-tune properties of the sensor, such as brightness or sensitivity, and also its affinity for calcium. So here, Janelia Fluor 635 was the fluorophore with which the sensor was developed. But by using more open dyes, we can have a sensor that shows a lower dynamic range, but that is brighter. And on the opposite, by using more closed dyes, we can have sensors that are dimmer, but have also a much less fluorescence in the resting states, which can be useful to adapt it to different applications. So finally, we tested this sensor in a cellular culture context in neurons that express cytosolic halocamp, to which we then delivered Janelia Fluor 635. And the sensor also works in neurons with a delta F over F of about 13% in response to a single action potential. The small molecule tunability is conserved in a cellular context. So by using different dyes, we can achieve higher sensitivity, for example. And in comparison, the sensor is much more sensitive than the genetically encoded NeoGecko-1. In terms of the kinetics property, the sensor also compares favorably in both the on and the off kinetics in comparison with NeoGecko-1. So so as a conclusion on this, what my previous work on hybrid calcium indicators has told us is that fluorophore tag pairs, and particularly rhodamine, can be used to substitute fluorescent proteins in the design of biosensor, and that can lead to achieve properties such as far red emission with high brightness that are still very hard to achieve by using fluorescent proteins to date. The sensor also has the very interesting property to be highly modular without the need to re-engineer the protein platform and can be used with diversity of small synthetic dyes. Finally, this sensor, which is a thing that I'm really interested in, is that it is also a very generalizable design. So in parallel to our work, the lab of Kai Johnson also developed hybrid calcium indicators based on a similar concept. And also at Janelia, Ahmed Abdel Fattah and Eric Schreider's lab has also in parallel shown that this general design could be adapted to the development of voltage indicators. So at this point, so currently the work on halocamp and hybrid calcium indicators is being pursued by Ellen Farrens in Eric Schreider's lab at Janelia, who is taking the task to push them to the next stage and implement them for applications in vivo. And so I started my group at EMBL. It's been now 1 year. And although it's still early days, I still wanted to introduce the group and the different directions that we are taking at the moment. And so yes, I was very interested into exploiting this hybrid approaches for biological imaging and how these pieces together could result in properties that cannot be achieved separately. And there are two main directions that we're working on at the moment. One thing that we're exploring is the design of photo-switchable, photo-activatable hybrid probes, in which we're exploring different type of new mechanisms that can lead to dye turn on and make them work in a way that they're activatable only when partner with their protein tag to circumvent certain level of backgrounds. And this is really aiming for applications in super resolution microscopy. And this work is led in my group by Bego and Francie, who just started as the first PhD student. And the second big area that we're starting to work on is photoacoustic imaging. And this is a project that we're really starting in collaboration with the Prevedel group, in which the design of molecular rapport for photoacoustic imaging requires molecular probes that absorb strongly at very far red wavelengths. So we're starting different projects in this direction, and this is led by Alex, who just started in the group as well. And and I'm really looking forward to showing you some new results in these directions, hopefully in the near future. So with that, I would like to finish up by thanking my group, of course, and our collaborators at EMBL, and also everyone at in the latest lab and Schreider lab, particularly everyone that contributed to this projects on calcium indicators that I've shown today. And thank you very much for your attention.
Yeah, thank you very much, Claire. Very exciting talk. I'm sure there are questions from the audience, and please, I want to remind everyone to use the raise hand sign if possible. So to give everyone a little bit a chance to think about questions, I have one for you, Claire. It's probably a very obvious one, which is that in many of the sort of biological demonstrations, you work with cultured neurons. I was wondering to which extent it's challenging to also get those hybrid sensors to work in animals. Yeah. Like
yeah, I think that's one of the challenges at the moment, which is that with all the benefits of moving to this two pieces approach, it also comes at the cost that you then have to deliver this small molecule dye. So the one of the main things is that this small rhodamine fluorophores by themselves can be very well delivered into living animal. They've been used in mice, in lice, in fish. So the small compound can be delivered. However, yeah, there's definitely certain challenges in making the sensor really capture the small molecule and label it. And there is some improvements to be done in this direction in terms of delivery, capture, and all this, which is one of the things that Ellen is pursuing at the moment.
Okay, yeah, thanks. Eduardo has a question. Please go ahead and unmute yourself.
Okay, thank you, Robert. Thank you, Claire, for the nice talk. Actually, on the same line of Robert, perhaps it's a naive question, but what are the direction you think are needed to develop better, let's say, delivery system for this dyes in either in animals or in whole tissues, as to use them in a as versatile way as live dyes, for example? Thanks.
Yeah, so I mean, it's a tricky problem because it is certain that very small structural changes in fluorophore structures definitely impact very much how these things are uptaken in animals or even in cells, that they change very much the behavior of this. So I think that identifying good dye structure that have this property of being very permeable and penetrating well, injections are ways of delivery for sure, but it's much better if we can find already some good fluorophore that have this ability to be uptaken quite well. And then we can work on genetically engineering the rest of the part, which is the easiest part for development. And then there's certainly other areas for small molecule deliveries using kind of like vectors, encapsulation, and things that can release then and serve as kind of like little transporters. Yeah, and I don't know so much about those.
Okay, yeah, thanks. Pratik?
Hi, Claire.
Hey, Pratik.
My question is, like, so do you think you can extend this approach to also include, like, let's say snapdragon dyes to have like to image two different modalities at the same time?
To swap with what thing? Sorry, I didn't hear you.
So if you want to measure calcium and something, another modality, so to go from imaging one thing at a time to like imaging two things at a time, do you think one can engineer like a orthogonal tag, like snap tag also?
I think there's really a very big need for this. It's the halo tag works really well for a ton of things, and it's a bit frustrating to see that on the dye side we've like solved multiplexing for a while now, but we're just missing more methods to actually label them and use them. So I think that, yeah, definitely the development of orthogonal tags is super critical for this. Yeah, I think that the halo tag as starting point is a really good platform because it's just been so well engineered that it's easier to restart from a system that works really well to then complexify it. But yeah, no, and I think that there is a lot of ongoing work in making better tag, more tags.
Thanks.
Cool. Alison.
Claire, thanks for your talk. So one thing that I've always been really curious about with this, when I first read this paper, is the fact that, you talk about replacing the dyes, but it also turns out that replacing the dyes changes the affinity for calcium, which seems to sort of challenge the idea that this is fully modular. So like, what do you think that the mechanism for that really is? Because based on the image of the crystal structure you showed, it seems like the calmodulin domain and you know, your whatever peptide you used are relatively far away from the dye. And so then how is the dye affecting that affinity? Or is that maybe not exactly what you're titrating that experiment anymore?
So I don't know. That's a really good question. There are two parameters that, well, I think that changing the affinity can be a feature in certain cases, you know. But no, I think that that indeed there's two things. So the affinity is a really good point, and also another thing that we've observed is that kinetics are also changing depending on the dye, which for me are two surprising things, because open closed equilibrium itself should not be so much the limiting step of any of that. So yeah, so I don't know. And as you're saying, it seems like the two domains are quite far apart. So I think that direct interaction of the dye directly at the calcium binding site to fit affinity would also be very surprising. The current, I mean, although we don't really know for sure how all this works for now, it seems like the working model is that it's actually propagation through the linkers that then trigger this open closed equilibrium because they're so far apart. So yeah, I mean, short answer is I don't know, and it is really surprising. But yeah, I think it's really interesting how the dye does affect this kind of properties that we would not expect to.
Brian, please.
Sorry, that does not answer your question at all.
Really super inspiring talk as always. I have a question about the photoacoustic sensors. I mean, I've always been super fascinated with this technique. Can you share a little bit? Is it also rhodamin based, or is it you have to switch the scaffold completely?
No, so we're definitely planning to start with rhodamin based systems. So I mean, that was our first approach. That what was very interesting to me is that in all this work and effort that was, you know, principally led by Luke in all this work in improving the brightness of this rhodamin, there's been a ton of terrible rhodamines made that are completely non-fluorescent, but still display very high absorption. So my general approach for this was to kind of take advantage of all this fluorophore improvement byproducts to engineer broken rhodamines that can be all non-fluorescent, but still very highly chromogenic, and repurpose them into this type of applications. So that yeah, and we've shown that we can indeed, by introducing some quenching groups, have a far red completely chromogenic rhodamines. So we're working on improving them as contrast agents, and then engineering sensors also based on this.
Great, thanks. Are there any other questions at this point? If not, then if I can ask a last question, it's a bit more for just my understanding is sort of coming not from the same field, is sort of what drives the brightness of the fluorophores and especially the rhodamines. You had this plot at the very beginning that showed brightness as a function of wavelength, and you said that, you know, the rhodamines, among others, are much more bright and more efficient in the infrared. So do you have some sort of structural understanding of why that is, sort of photophysical understanding of why, and can that drive the design of even sort of more improved redshifted sensors?
Yeah, I mean, it's certainly also true with synthetic dyes that the more you go into the far red, the more making super bright stuff is difficult, because as to shift the wavelength to the far red, you have to extend electronic conjugation. So you do have to complexify the scaffold, and this also leads the way to a variety of different accessible processes for non-radiative de-excitation. So you do have more and more competition from processes that release energy without emitting any light. But I do, yeah, indeed, there's a much better understanding at the synthetic dye level of those different type of quenching mechanisms and competitive processes. But and more than this, there's also very much this whole synthetic chemistry dimension that you can access to actually make this small modification that will prevent one process to favor another. And this is thing that engineering those minor modifications at the level of a fluorescent protein chromophore are extremely complicated.
Okay. All right, thanks. I see no further questions. So unless there are any last minute takers, then yeah, let's thank Claire again for a wonderful talk.
Thank you for having me.
And I believe, Ulrike, then the last point on my agenda is just to basically give an outlook for the next speaker, who will be Stefan Saalfeld, who will be speaking on January 12th about his work in image registration and reconstruction of electron microscopy, I guess, data sets, right? Anything else I should add, Ulrike? Or?
So maybe only that we actually plan to extend the series or have an hopefully infinite combined EMBL Janelia series. So currently the talks are planned until February, but be prepared for more exciting sessions.
Yes,
yeah, until we run out. Pardon?
And I were already writing to each other about potential speakers. So we have a few lined up. I hope you too and Janelia. I think so far it's been a great series, and I would be very much for extending it further.
Yeah, so hope to see you all soon at the next series, and thank you very much for coming today, and have a nice Christmas.
Yes, happy holidays to everyone.
Okay, bye-bye.
Happy New Year. Bye-bye.
Thank you.